Presentation will be given by Dr. Alice Manzewa, University of Western Cape from South Africa. Over to you, Doctor.
Thank you. I'm just going to share my screen and then I can start with the presentation. Can you see my screen with the slides?
Yes, Doctor.
Thank you. Okay, my name is Alice Mwanjiwa Kanyerere. I am a biotechnology master's student at the University of the Western Cape located in South Africa, so not yet a doctor. I will be presenting on my master's work, which is titled The Morphophysiological and Biochemical Characterization of Amaranthus caudatus. Drought stress is essentially one of the biggest problems that we have because plants are sessile organisms, which means that they are unable to move from the environment should the conditions change. This makes them susceptible to different types of stresses such as abiotic stress and biotic stress. Abiotic stress is defined as the adverse conditions that plants face caused by non-living factors such as heavy metal stress, high salinity levels in soils, unfavorable temperature conditions, and whether it's be extreme cold or extreme hot, and as well as nutrient toxicity and drought stress. Drought is a form of abiotic stress, which is caused mainly as a result of climate, which leads to long-term periods without significant rainfall. This leads to inhibitions of plant germination. It reduces the crop yields. It decreases the germination parameters. It reduces the photosynthetic rate, thus shortening the grain filling period. According to literature, drought alone is responsible for a substantial reduction in crop yields, which negatively affect the physiological, biochemical, as well as the molecular processes in plants. So on the screen, I'm just showcasing the different effects that drought has on crops. The first image just showcases the effect of drought stress on a maize field. The second image showcases the effect of drought stress on an amaranth field. And then on the next slide, I am just showcasing the different types of effects of drought stress, just grouped into two categories. So you've got the one side shows the main effects of drought stress and how plants are able to combat them. And then the next one shows the main photosynthetic drought stress responses, where the plants will either reduce water intake or they'll conduct stomata closure, or they'll increase the rose scavenging substances such as sod pods, cat, and APX. Seed germination is one of the most important factors in a plant's growth because it is at this point where the plant will either remove itself from the seed dormancy stage or it will remain in the seed dormancy stage until the environmental conditions are favorable. This complex adaptive process of germination in higher plants is often regulated by internal factors and external factors that the seed would be exposed to, which makes it very important for the environmental factors to be favorable so that the seed can be able to germinate. Water deficiency, which is experienced during germination, can cause changes in the enzymatic activity and reduce hydrolysis to protect the seeds. So enzymatic activity changes, we're talking of the amylase activity. And we see all these effects as the unsynchronization of the seedling images, the poor uniformity in plant growth, which have been observed as some of the direct effects of drought stress on both amaranthus, rice, and quinoa. Amaranthus is a genus which belongs to the Amaranthaceae family, which is native to South America and Asia. There are more than 70 species in this specific family. However, scientists are now suggesting that there might be over 150 species. This family is categorized into three categories. You get grain amaranth, you get vegetable amaranth, and you get wheat amaranthus. It is an underutilized indigenous crop, which is highly nutritious and it is rich in antioxidants. This makes it perfect for human consumption. Vegetable amaranth is a popular food in the Asian continent in countries such as Bangladesh, Pakistan, and India due to its low cost, its lucrative color, the taste, and the abundance that it has in nutrients. It is often grown throughout the year as well as in the gaps of the foliage between winter and summer. And it is also said to be highly tolerant to temperature changes, to drought, pests, as well as diseases. So amaranthus itself can also be used to eat. For example, you can eat it as cereal, or you can crush the seeds up and then make flour to make cookies, or you can eat it in soups as well as in salads. The figure on the side just showcases the different categories of Amaranthus. So in the grain Amaranthus, you get Amaranthus caudatus. In the leafy Amaranthus, you get Amaranthus tricolor as well as Amaranthus viridis. And then in the wheat type Amaranth, you get Amaranthus spinosas. The next image just showcases the different types of species you'd find under grain Amaranthus. So as I mentioned, you get Amaranthus caudatus, you get Amaranthus hypochondriacus, as well as Amaranthus cruentus. I will be solely focusing on Amaranthus caudatus because this is one of the few species that is said to be drought tolerant. However, the mechanisms in which it uses in the plant systems to induce this drought tolerance is not completely researched. So I'll be looking at the 3 genotypes of Amaranthus caudatus, namely love lies bleeding. I'll be looking into red garnet as well as ponytail. The main aim of this project is to assess the different effects of drought stress on the germination of various Amaranthus genotypes and its response mechanisms. And this will be done, first of all, by germinating Amaranthus caudatus under both well-watered and water-deprived conditions. And then I will assess the morphophysiological responses of Amaranthus caudatus under both well-watered and water-deprived conditions. And finally, I am going to be looking into the biochemical response of Amaranthus caudatus in response to the well-watered as well as the water-deprived conditions. So it is said that by the year 2050, the global population will reach between 10 and 11 billion people. And this is putting an explosive amount of pressure on the food systems that we have, particularly in continents that rely on agriculture as the main source of food, such as Africa and Asia. And to be able to achieve a sustainable food security for this expanding population, we need to explore resilient, drought-tolerant crops that can thrive in harsh conditions, such as Amaranthus caudatus. So not only is Amaranthus caudatus highly nutritious, but it also offers boasting protein-rich seeds, which have been parts of the human diets for centuries. It can also be a critical trait that we can use in modern agriculture as we're all trying to find seeds and plants that are able to induce drought tolerance with the increase in climate change conditions. By understanding the biochemical and physiological mechanisms in Amaranthus caudatus particularly, we can harness the potential of this crop to be able to help us solve the food security issue, as well as be able to induce some of the mechanisms that we will find from this crop in other crops to induce the same. The whole idea of this project is to first of all conduct a relationship or strengthen the relationship between farmers and scientists alike, as farmers possess some of the traditional knowledge regarding the plant itself. And then by expanding our understanding on the Amaranthus and how it responds to drought stress, we can effectively develop strategies to mitigate the different challenges that we're facing worldwide. And finally, the study will serve as a gateway to comprehending the behavioral patterns of different Amaranthus species and of varying levels of drought, specifically within the South African climate. So this study itself is divided into 3 drought treatments. We've got T1, T2, and T3. T1 is a drought treatment that is induced over 7 days. T2 is a drought treatment that is induced over 14 days. And T3 is a drought treatment that is induced over 21 days. For the sole purpose of this presentation, I will be giving the results from the preliminary data that is concluded after performing the T1 treatment on Amaranthus genotypes. The plants were first germinated in a green tunnel for 21 days to establish the seeds. Afterwards, water deprivation was induced from day 21 to day 28, and then the seeds were taken from the tunnel to the lab for analysis. In the lab, the very first analysis that we performed was the morphological assays, where we looked into the number of leaves, the shoot length, the plant biomass, and the relative water content. This would give us an idea to the water use efficiency systems that are found in the 3 different Amaranthus caudatus genotypes. Then we looked into the physiological assays, particularly germination percentage. So germination percentage will give us the speed as well as the seed viability of the genotypes per the containers and the wells that they were grown in. And then we looked into chlorophyll content and carotenoids. So chlorophyll content and carotenoids, these are for the synthetic pigments that are supposed to protect plants when plants are under drought stress. So we're monitoring the different amounts depending on the drought stress that we have versus the control that we had. Finally, we looked into the biochemical assays, which were divided into two groups. So we were looking into the osmolyte content as well as the oxidative stress markers. For the osmolyte, we looked into proline content and soluble sugars. And for the oxidative stress markers, we looked into the malonyaldehyde content as well as the hydrogen peroxide content. And these would first of all give us an idea of how the plant is performing under the stress that is induced in terms of, is it going to use the osmolytes to induce some sort of osmotic protection method, which we call the osmoprotectants, or is it going to give us signals in the oxidative stress markers to say that there is a lot of accumulation of the reactive oxygen species, or just signaling the plant to use another mechanism to induce the drought tolerant trait. The first results that I have are from the germination parameters, which is particularly the germination percentage. So in the germination percentage, we can see that red garnet on day 7 had the highest germination percentage in both the control and the drought treatment when we compare it across board with the other genotypes. And when we look into love lies, we see that day 3, 5, and 7 showed a consistent increase, which means that the plant wasn't stressed per se, but the seeds were viable and they were growing and germinating slowly. And ponytail had the highest error bars, which means there was not a synchronized kind of emergence, but then the seeds were still viable. When we look into the plant biomass, the fresh weight particularly, we can see that love lies, the drought treatment of love lies showed a significant increase. The ponytail also showed an increase, though it was not significant. And red garnet, again, across board, had the highest fresh weight in terms of the drought treatment when compared to the control. Looking into the dry weight, we also see that love lies had a significant increase in the drought treatment, and ponytail also exhibited a significant increase in the drought treatment as well. But red garnet, once again, was able to outperform all the 3 genotypes. Finally, looking into the relative water content, we can see that love lies particularly had a significant increase in the drought treatment than the rest of the genotypes. But similar to the first 2 results, red garnet had the highest relative water content across board to all the genotypes. So based on these results alone, we can safely say that red garnet seems to induce its growth parameters to be able to induce the drought tolerant trait when compared to the other 2 genotypes. Then I looked into the photosynthetic pigments of the different genotypes, and this is where the results got a little bit interesting. So for the first one, we found that ponytail was able to exhibit the highest number of leaves in a plant as compared to the other genotypes under the same conditions. And then red garnet induced a higher and more significant shoot length and chlorophyll A when compared to the other genotypes. And then we looked into love lies bleeding, which showcased the highest chlorophyll B content when compared to the other genotypes. And finally, we looked into the total chlorophyll and carotenoids of the 3 genotypes, and it was found that red garnet once again outperformed the other genotypes by giving us the highest content of the total chlorophyll and the carotenoid content. Based on these results and the previous one, we can safely say that red garnet seems to outperform the other genotypes in terms of the growth parameters when it comes to inducing a sort of drought stress tolerance mechanism. Finally, I checked the osmolyte content of the 3 genotypes. And first of all, I looked into the proline content. So proline accumulation usually occurs when the plant is under a lot of stress, because this is one of the first traits and one of the first signals of the plant trying to induce either drought tolerance or showcasing that the plant is actively under drought stress. So looking into red garnet, the drought treatment of red garnet had a significant increase. And the ponytail genotype also showed a significant increase in the drought treatment. However, the love lies did not show a substantial increase between the control and the drought. Then when we look into the total soluble sugars, which is also an osmolyte that plays a similar role to proline, we can see that love lies had the lowest total soluble sugar content when compared to the two genotypes. But the biggest difference here is in the red garnet, you see a substantial decrease in the drought sample, as well as in the ponytail where you see a substantial decrease. However, in the love lies, even though it has the lowest water soluble sugars, you can see that there is an increase in the drought sample. So perhaps more research can showcase that maybe love lies uses water soluble sugars as a drought stress tolerant trait, while the other two genotypes use proline as their drought stress mechanism. And then finally, I looked into the oxidative stress markers. So with the oxidative stress markers, I started first with the hydrogen peroxide content. We showcased that love lies had the highest hydrogen peroxide content under the drought sample. And in the ponytail, it showed a significant decrease in the drought treatment than in the control. So based on this results, we can say that perhaps love lies was able to induce a better signal with the hydrogen peroxide content than the other two genotypes. And finally, I looked into the MDA content of the plant, where I saw that the ponytail had the highest significant increase in the drought sample as compared to the rest of the genotypes. In conclusion, this study demonstrated that Amaranthus caudatus can withstand up to 7 days of water deprivation without exhibiting significant oxidative damage or impaired growth under greenhouse conditions. And in terms of the three genotypes, this study showed that red garnet was outperforming all the other two genotypes because it had the highest shoot length, it had the highest photochlorophyll and carotenoid content, and it also exhibited a significant increase in the proline. So perhaps just for the 7 days under the climate that we're in, red garnet is the best genotype to plant when these conditions are met. However, T2 and T3, when performed, will show us adequate results on which we can conclude which genotype is best to plant in a climate such as South Africa, particularly in the Western Cape, which receives less than 400 millimeters of rainfall each summer. Further research can be done into exploring the drought tolerance mechanisms of Amaranthus, which can be used to inform us of the strategies that can be used to improve the resilience in other crops and provide sustainable agriculture practices. And additionally, it can also offer innovative solutions which are crucial to mitigate the adverse effects of climate change on food production. I would like to acknowledge the University of the Western Cape for providing the platform for me to conduct my analysis, as well as my funders, the ARC, the Water Research Commission, and the National Research Foundation. And I'd also like to thank my lab, the Molecular Sciences and Biochemistry, for accepting the invitation for me to perform and to present my results to this conference. Thank you.
Thank you, Dr. Alice, for your valuable presentation. Now the session is open for questions. Any questions for Dr. Alice? Does anyone have a question? Okay, doctor, I think there is no questions. I'll share your email ID in the chat box. If anyone have a questions, they will contact you through the mail. Thank you so much. We'll move to the next presentation. The next presentation will be